Hello, good afternoon. We are going to start the session in a couple of minutes. I see that many people are joining right now, so we will start in a second. Thanks. Hello, welcome. I see that more people are joining. So as I said, we are going to start in a couple of minutes. Thanks for joining, and I just want to wait to give the opportunity to everyone to join. Good afternoon, everyone. More people still joining. We are now, this is 3 p.m. already, so I'm going to wait just a moment for someone else to join, and we are ready to start. Okay, so let's go. Again, welcome everyone, and thank you for being here today. My name is Noelia. I am a scientific content marketeer at Encardia, and I'm going to be the host today. I'm very happy to be hosting this session because it's actually the third one that we have dedicated to the neurodegenerative diseases. And we've seen that this topic is of great interest in the audience. So that's something very, very good to know and to see that you like and enjoy these webinars. So before we go with the science today, I will make a brief introduction about Encardia for those of you that still don't know us. And so Encardia has a great expertise in the use of human-induced pluripotent stem cells, and we offer different solutions and services to apply this technology into drug discovery. We go from iPS sourcing and reprogramming to cell manufacturing and differentiation of these cells into many different functional cell types. And the manufacturing process can be performed at the scale needed, also larger scale. And we also offer services for assay development, disease modeling, and screening. And to give you a more concrete idea of our capabilities, you see here in this slide that we have developed established processes for the differentiation of iPS cells into cardiovascular and neuronal models. And we can also develop completely new processes for other cell types. We generate custom disease models with different methodologies, and we ensure that these models express the disease phenotype that you are interested in. And then we have a wide range of in-house assays that can be performed in high throughput, and they evaluate different functional properties of your cell models or metabolism, cell viability, and also biomarker expression. So this gives you a very good idea of the safety and efficacy of your compounds. And basically, at Encardia, we truly believe in the benefits of human iPS cells for drug discovery, because first you can derive the cells from a single source, so you are getting more consistency and reproducibility. You can also create disease models that recapitulate the disease phenotype that you are looking for or that you are studying. And because these models are generated with human cells, this means that the translational power is very high. And this also involves that your in vitro screenings, the outcomes that you have from these screenings will have higher chances of success when you go to the in vivo testing. Therefore, our mission at Encardia is to use the human stem cell technology to help bring in better therapies to patients faster. And now going back to the webinar and the reason that why we are here today, here you have an overview of the other previous webinars that we had dedicated to this topic. They are both available now on demand on our website in case you missed them. And today we will be talking about and we will be presenting a model for Alzheimer's disease that was generated with our human iPS-derived neuronal and glial cells. And although this is the last webinar dedicated to this topic, there are more things coming. Next month, we will have a webinar dedicated to cardiac safety, and the next one, atrial fibrillation, and more. So we will keep you posted. And the presenter of today is Dr. Laura Callo. She has extensive experience in developing functional assays and iPS-based models for the study of neurodegenerative diseases. And she has been collaborating with companies to develop new drugs for these diseases. And now she is a senior neuroscientist at Encardia. And unfortunately, due to personal reasons, she can't be here live today, but she kindly pre-recorded the session for us, so we can still enjoy the presentation. And then for the Q&A session, we invited another two neuroscientists, Shui and Isabel, and they will be here live after the presentation to answer all your questions. So regarding the questions, you can see in the right side of your screen that there is a chat option. So you can type everything, all your comments and questions there during the presentation. And at the end, I will address them to the scientist. So let's start. I will launch the video and I'll see you in a bit when the presentation is finished.
Good afternoon. Thank you for joining this webinar. Today, I'm going to describe the development of a model that recapitulates pathological cellular features of Alzheimer's disease using human-induced pluripotent stem cells. 55 million in the world, 55 million people in the world are living with dementia. And by 2050, this number is unfortunately predicted to increase to 139 million. There is really a lack of disease-modifying therapies, and model systems that really robustly mimic human AD for drug development are crucial for translation. Alzheimer's disease and other neurodegenerative diseases are characterized by tau hyperphosphorylation and aggregation. Tau in physiological condition binds to microtubules and stabilizes them. When tau becomes hyperphosphorylated, such as, like for example, in Alzheimer's disease, microtubules fall apart, tau aggregates forming neurofibrillary tangles, as you can see here in this immunohistochemistry, where you see a neuron burdened by tau in the cytoplasm and in the neurons. Besides Alzheimer's disease, other neurodegenerative diseases are characterized by tau inclusions, and these are amyotrophic lateral sclerosis or frontotemporal dementia and many other neurodegenerative diseases. So the cellular dysfunctions that have been described in human neuronal models of AD are several. Amongst these, we let's say cite tau aggregation, tau hyperphosphorylation, autophagosome and lysosome impairment, mitochondrial dysfunction, neuronal loss, abnormal morphology of neurons such as short neurites, varicosities, and of course activation of the resident immune cells in the brain, microglia and astrocytes. So our aim, or we wanted to use generate a physiologically relevant model of tauopathy using Encardia human iPSC cortical neurons. And for that, we engineer these neurons to express an untagged 4R tau scaffold and promote aggregation following exposure to tau seeds derived from Alzheimer's disease cases. The experimental paradigm is summarized here. Briefly, we add our tau seeds at 10 days, wash out, and allow neurons to grow for 10 more days, 10, 11 more days. The readouts for this experiment is high content imaging, and then we measure phospho-tau panta, we measure autophagy and neuronal loss. So in this model, we said that we really wanted to focus on using tau seeds from human disease cases. And these cases are very heterogeneous, of course. The seeds are heterogeneous depending on the age, sex, and area of the donors. And therefore, we use 6 disease cases, 1 non-disease individual, and unseated neurons as controls. This, of course, was, let's say, allowed by agreement with the Netherlands Brain Bank. And as you can see, amongst all the samples we tested, we found that disease case number 4, which is an Alzheimer's disease case, induced a 4 to 5-fold increase in the number of 88 panta, whereas other cases induce a less pronounced phenotype. Phenotype can be observed here, and you can appreciate the difference between the unseated neurons here on the top or the seated neurons here, where you can see